Today we will discuss about red cell indices and osmotic fragility. Now, what are the red cell indices? We had discussed about packed cell volume, hematocrit in the previous class. We had mentioned a word about red cell indices also. Here we'll discuss red cell indices in detail. So these indices, they show the health status of a person. Okay? There are MCV, MCH, MCHC. We'll discuss one by one in detail. First one is mean corpuscular volume, abbreviated as MCV. So MCV, it is basically average volume of each RBC. It can be calculated by this formula, hematocrit expressed in percentage divided by RBC count in million per microliter multiplied by 10. Normal value of MCV, it is around 87 femtoliter in both males and females. When MCV is normal, we can say RBCs are normocytes. Okay, when it is more, they are macrocytes. When it is lesser, we can say these are microcytes. Volume increased or volume decreased. Second index, it is your mean corpuscular hemoglobin, abbreviated as MCH. So it represents the hemoglobin concentration in 1 RBC, and it can be calculated by the formula given here, hemoglobin expressed in percentage divided by RBC count in million per microliter multiplied by 10. Now the normal value of MCH, it is 29 picogram in both males and females. Next index, it is mean corpuscular hemoglobin concentration, abbreviated as MCHC. It represents average hemoglobin concentration in 1 RBC. And the formula for calculation is here, hemoglobin in gram percentage divided by hematocrit or PCV in percentage multiplied by 100. Normal value of MCHC, it is around 34 gram per deciliter in both males and females. So whenever MCHC is normal, we will say RBCs are normochromic. When it is lesser, we will say hypochromia is there. But there is very important question, anemia can never be hyperchromic. Okay? MCHC, it is never beyond 38%, as these cells, they cannot hold hemoglobin beyond the metabolic limit of the cell. It is a very important reasoning based question that is asked in examination, why anemia can never be hyperchromic. Okay, so we had talked about MCV. Normal MCV means normocyte, decreased MCV microcyte, more MCV than normal macrocytes. MCH, MCHC. When MCHC is normal, normochromia is there. MCHC, when it is lesser, there is hypochromia. But anemia can never be hyperchromic. Now, what is the significance of these red cell indices? Why do we need them? It is for the laboratory diagnosis of anemia. Okay, if the person is suffering from anemia or not. For the differential diagnosis of anemia. What do you understand by differential diagnosis? We have made a diagnosis, person is suffering from anemia. But anemia is of several types. It can be because of deficiency of iron, could be because of deficiency of vitamin B12, folic acid deficiency, could be sickle cell anemia, could be thalassemia, could be secondary to some liver diseases, secondary to some kidney diseases. So for the differential diagnosis, which type of anemia the person is suffering from, we require red cell indices. For example, in case of iron deficiency anemia, there is microcytic hypochromic anemia. Means microcytic, MCV is lesser, hypochromic, MCHC, it is also reduced. Okay. For the classification of different types of anemia, we had just discussed this. which type of anemia the person is suffering from. For the classification, we'll discuss about the classification when I'll teach you anemia in detail. Now about osmotic fragility. In physiology, everything lies in the name. Osmotic, related to the osmotic changes. Fragility, how much fragile a thing is. So the ease with which the RBCs, these are broken down or get hemolyzed when these are present in the hypotonic. It is not hypertonic, it is hypotonic. Okay. So normal values, fragility, it starts when the cells are placed in 0.48% NaCl and it ends when the cells are placed in 0.34% NaCl. So these are placed in the hypotonic solution, hypo, when the osmolality is lesser as compared to plasma. So solutions that are isotonic with plasma, 0.9% NaCl, 5% glucose, 10% mannitol, 20% urea. So when the cells are placed in hypotonic solution, hypotonic, 0.48% NaCl. Okay. RBCs, they get swell up and ultimately they can burst. Why can they burst? Because solution outside is hypotonic. Water will, or the solvent part, it will try to enter into the cell. By the process of osmosis, RBC will get swollen and ultimately they can undergo bursting also. But if the cells are placed in the hypertonic solution, they will undergo crenation. As you can see here in the first diagram, you can see when the RBCs are placed in the hypertonic solution, there is movement of solvent out of the RBC into the solution. They will undergo shrinkage. Second part, it shows when the RBCs are placed in the hypotonic solution, there will be imbibition of water. Isotonic is normal. Okay. So osmotic fragility, ease with which the RBCs are placed, RBCs are broken down or hemolyzed when these are placed in hypotonic solution. They tend to swell and ultimately burst down. Thank you very much for this. In the next class, we'll start with hemopoiesis, erythropoiesis and its regulation. It will be taken up. Thank you very much.